Today I'm going to be telling you a little bit about some of the work that my lab has done that is focused on the question of sensory prediction errors in neocortical microcircuits. And I think this is interesting from a neuroprosthetic standpoint because what I'm going to push towards in this talk is the idea that there might be two, if you will, targets for neuroprosthetics researchers to think about as targets that they should actually be striving to record from and or stimulate in the brain that they might not otherwise think would be things for them to be concerned with. So I want to start this with an argument for the idea that there is a general principle of intelligence that may have informed many of the operations of neocortical microcircuits. This general principle is the idea that one of the ways in which you can learn a good representation of the environment in order to mediate decision-making and behavior and all the important things you have to do is to learn a model of the world by predicting the next set of sensory inputs you're going to receive based on the past set of sensory inputs you've received. The reason that this is a potentially general principle is that in order to predict the next set of stimuli that you're going to receive, really, arguably, you need to have an internal model of the world that captures the structure of the objects and how they move through the world effectively. So for example, if you see a frog, in order to be able to predict what is going to happen when the frog jumps, you need to understand something about the nature of its legs, how its legs give it propulsion, how gravity impacts its body, etc. So if you can actually predict the future visual inputs that you get, say, when you first see a frog start to jump off, chances are that internally in your brain, you've got a reasonably good model of the physical world. And indeed, we see in artificial intelligence that there's been great success recently in developing AI systems wherein the ability to predict stimuli or alternately to predict modified stimuli from one another is in fact a good means of developing structured representations that can serve to form the basis of future learning. So interestingly then, given that this may be a general principle, as I argue, there is indeed a huge body of work in neuroscience showing that the brain responds very strongly to stimuli that violate expectations. So if you structure the stimuli in such a way such that an animal or a person would reasonably make some prediction about what they're going to see next or what they're going to hear next, whatever, and you then violate that expectation and you present them with something that doesn't match those expectations, you see large responses in the brain. You can see this in EEG from scalp recordings, you can see this in fMRI, and you can see this in two-photon imaging, which is what I'm showing here. So this is a paper from Holman et al. That was two-photon imaging in layer 2-3 of mouse primary visual cortex. They had mice on a little running wheel here. I assume you can see my mouse. Someone please let me know if not. So they had a little running wheel here, and the mice would be presented with visual stimuli on a screen. And what they presented them with were sequences of random Gabor patches that were repeated multiple times over. So there was a specific sequence that they always followed, A, B, C, D, E, A, B, C, D, A, B, C, D, etc. And then what they would do is occasionally insert unexpected, previously unseen random Gabor patches into these sequences. And what they reported is that activity in layer 2-3 cells showed this deflection upward in response to the novel stimuli, the unexpected violations, and not in response to the expected stimuli. And this fits with a whole host of models, sometimes referred to as predictive coding, but there's a broader class beyond the specific model that many people think of when they think of that. What I really just want to emphasize here is the idea that, what we know is that the brain shows very salient responses to stimuli that violate expectations. Now, in this data that I'm about to show you today, we wanted to answer 3 unresolved questions about these responses. So when the brain responds to a stimulus that violates its expectations, what's going on in the structure of the neocortical microcircuit, which is what I've got illustrated on the right here. So most of you probably already know this, but just a brief overview for those who don't. When we look at the neocortex, we see this laminar structure that's typically broken down into 6 layers, though layer 2, 3 often get treated as basically the same. And within each layer, you have different cell types, and each of the different cell types has different targets that project to different parts of the brain and receive inputs from different parts of the brain. And so within this microcircuit, there are a variety of unanswered questions about the responses to unexpected stimuli. And some of the questions that have been unanswered, which we want to address here, are first, what's happening in the apical dendrites of pyramidal neurons? Now, this is interesting for us because, so the pyramidal neurons in the neocortex are the most common form of neuron, and they are likely the workhorses of information processing and behavioral control in the neocortex. And they have these long dendrites that reach up towards the cells in layer 2, 3, and layer 5 anyway, have these long dendrites that reach up towards the surface of the brain, the peel surface here in layer 1. And these dendrites have a variety of interesting properties, one of which is that they are one of the principal recipients of higher order information from other brain regions. So from associative brain regions, both cortical regions and thalamic regions, these apical dendrites receive top-down signals. And this is interesting when we think about this question of stimuli that violate expectations, because for a variety of theoretical and practical reasons, it's entirely likely that higher order associative areas are responsible for forming some of these predictions. And that the information that they provide top-down is part of what would allow the circuit to detect violations of expected stimuli. Now, another question that we wanted to ask is what is happening with other cells in the microcircuit that can project up the hierarchy? So a lot of the work that's been done in animals has been focused on layer 2, 3 cells. And that's been in part just because they are optically accessible for 2-photon imaging. But layer 5 pyramidal cells are also a very interesting target because they can also project up the cortical hierarchy and communicate with those layer 2, 3 cells, and they have these apical dendrites. And people don't really know what happens in layer 5 cells when these unexpected stimuli occur, so we wanted to address that as well. And then lastly, we wanted to understand what happens over the course of time. So if you encounter some unexpected stimulus and then you see it repeatedly again and again and again, what happens in terms of the changes in neural responses over the course of these repeated exposures? So to answer these 3 questions, we actually applied to the Allen Institute for Brain Science's Open Scope program. This is a program where, whereby computational neuroscientists can apply to have the Allen run experiments for you within the parameters of a particular set of experiments that they are capable of running. And if they accept your proposal, they'll collect the data for you, give it to you for first pass on analysis, and then it'll be open freely for everyone to analyze. So the data I'm going to show you today came from an open scope project that myself, Joel Zeilberberg, Joshua Bengio, and Tim Lillicrap submitted. And we are working on a paper now. And so once the paper is out, we will in fact share this data. And so all this data will be open for anyone else to analyze in the not too distant future. And I just want to recognize here the analyses that were done that I'm going to show you were done by a PhD student in my lab, Colleen Gillen. The entire project was really pushed forward by myself and Joel Zeilberberg, my colleague at York University, and his postdoc, Jay Pina, was also key to all these analyses, and Tim Henley, a student with him, also helped with some of this. Okay, so what did the Allen do for us? What was our proposal, basically? So we asked them to collect 2-photon calcium imaging movies from both layer 2, 3, and layer 5 pyramidal neurons in mouse primary visual cortex. Now, because one of the nice things about doing this in mice is that we can genetically target those 2 cell types. So the recordings were specifically just for layer 2, 3 cells or just for layer 5 cells. And then furthermore, what they did is they recorded both in the dendrites, the distal apical dendrites in layer 1, and at the cell body layers in layer 2, 3 and layer 5. So basically we have 4 different imaging conditions here in these experiments. And what happened, and the GIFs will start in a moment for this, is that they performed 2-photon imaging in awake behaving animals. The animals underwent 3 hours of habituation to get them used to the stimuli in order to develop, as it were, expectations. Now, of course, we can't know whether a mouse expects something or not. We can only say that they've been exposed to some stimulus repeatedly, and then we change the stimulus. So, you know, take the entire phrasing of expected versus unexpected with some caveat, or sometimes I use the word surprise. We don't exactly know what the mouse is experiencing, but we can say just that we had habituated them to a particular set of stimuli, and then we changed the stimuli to look at the responses. And when we changed up the stimuli, then we would do an hour-long recording session once every day for 3 days. So we have 3 different recording sessions, and we look at how the responses change over the course of those 3 days as the animals continue to get experience with these unexpected violations of the stimuli. So here on the left, you can see a mouse on the running wheel. And there on the right, you can see what some of the raw images look like from the apical dendrites in layer 1. So that little lightning storm there, those are the apical dendrites lighting up as the animal views visual stimuli. Now, the stimuli that we presented to them were similar to the ones that I just mentioned to you for the Holman et al. Paper. So we presented them with sequences of randomly placed Gabor patches like this. Now, the way that we structured the expected versus unexpected component of it was as follows. In most of the sequences that the animals saw, and during all of the habituation phase, we had 4 images of random Gabor patches, and for each image, the orientations were drawn randomly from a distribution with a particular mean orientation. Now, we changed that mean orientation every time, so it's not that the animals always saw the same orientation by any means. It's just that within a particular sequence, we call these images A, B, C, D typically. So the A, B, C, D images, the orientations were all drawn from the same distribution. Now, when we violated the animal's expectations, what we would do is we would replace the final D frame that had normally orientations drawn from the same distribution with a set of Gabor patches that had orientations drawn from an orthogonal distribution. So you can see that here in these stimuli. When the dot is green, these are the expected sequences. So all 4 images have the same orientation mean. And then when the dot turns red, what you'll see is that in the 4th frame, the Gabor patches all have a orientation drawn from a different distribution there. Okay, so the animals only saw those different distribution or orientation images, which we call the E images or the surprise images, during the recording sessions. And they only saw it 10% of the time, so as to maintain some of their expectations. Now, what we found in summary, because this is a relatively short talk, so I'm going to keep it brief, is that effectively we saw a very interesting distinction between how the somata and the apical dendrites responded in these 2 cell types. So what we found is that at the start of the imaging sessions, the somata, which are the blue traces down here, showed larger responses. So here I should just clarify this. On the x-axis we have time, and we have the A, B, C, and then in pink here, this is where we either present the expected D frame with orientations drawn from the same distribution, or the unexpected E frame with orientations drawn from an orthogonal distribution. And what we found was that when we presented the E frames, which are the colored traces here, the D frames are the gray traces, in the somata, we got larger responses to the E frames than to the D frames in the first imaging session. Now, this matches what Holman et al. Reported and what various other people have reported, and is in line with a lot of the stories that people have told about the nature of predictive responses and sensory prediction errors in the brain. But what we saw that was very unexpected was that, and I should say the other thing that we saw that was perhaps in line with existing hypotheses is that those responses in the somata died down over time. So as the animals experienced more and more of these E frames, the responses in the somata eventually came to be basically the same between the E and the D frames. So there was no longer this like unexpected surprise response in the somata. But what's interesting is we saw the reverse pattern in the apical dendrites. Whereas the apical dendrites did not respond to the unexpected frames at the start of imaging, they actually came to respond to the unexpected frames more than the expected frames over the 3 days. And by the end of the third session, we were getting very big responses in the apical dendrites to these E frames. Here you can see that broken down just as a difference. So we take the integrated delta F over F for the E frames, we subtract the delta F over F for the D frames. And then for statistical control, we do shuffled versions of those differences where we swap the E and the D labels. And what you can see is that whereas there are significant responses in the somata that are outside of the shuffled data zone in the first imaging session and not in the dendrites, by the third session that has reversed, and now we have something that's no longer statistically different from chance in the somata, whereas we have significant differences in the apical dendrites. Here's an example of the responses in the apical dendrites. So these are just delta F over F traces normalized between 0 and 1, where 1 is max and 0 is nothing. And this is also broken down across the different mean orientations for the frames. So on the top, we have the expected D frames, and on the bottom, we have the expected E frames. This is for one animal in imaging session 3, averaged across multiple exposures of the stimuli and in the layer 2-3 dendrites. And what you can see is that whereas there are some responses to the D frames, and there are, of course, responses to the start of the sequence, there are these incredibly large responses in the apical dendrites in response to the E frames in this animal. And we actually saw this in the other animals we recorded from as well. They weren't always this strong, but they were always very remarkably strong. And for those who have worked with mouse 2-photon imaging data, it's non-trivial to see big responses like this to a stimuli. It was very interesting for me to see how clear this was. And what's also interesting is that you see this response across all 4 orientations. So it's not even that the dendrites have just learned to respond to a particular orientation and that happens to be the surprise orientation. This is happening in response to just the unexpected E frames, period. Now, of course, I just want to say, by the way, the latency here, you can see that a big part of the response actually happens during this gray screen, but it comes on before. So it is in response to the E frame, though it's got quite a delay on it, which is interesting when we think about that top-down question. The other thing about this is that though there are responses to all the orientations, different cells respond to the different orientations as we'd expect. So as a result, the responses do carry stimulus information. So when we run a decoding analysis on the responses, we can decode better than chance, shown in gray here, what the orientation of the frames, the mean orientation of the frames were. Though, of course, that ability comes on as the responses in the apical dendrites come on for the surprise frames, and it gets lost as the responses to the surprise frames disappear for the somata, which is interesting. So basically, that information is being lost in the somata, but being carried in the apical dendrites. And lastly, just as a control, we checked whether these responses were ultimately being driven by behavior, because there's a lot of evidence that mouse visual cortex is, in fact, highly driven by motor inputs. And what we found effectively is that there were no statistically significant changes in running speed to explain these responses, and no statistically significant changes in pupil diameter, suggesting that this is not something driven by the animal responding to the thing, and then there being a response in visual cortex. It's driven by the stimuli. So to cap off, our data supports the idea that there are prediction error signals driven by top-down information potentially within different parts of the circuit. And in particular, it highlights the fact that the apical compartments of the circuit are doing something different than the somatic compartments. which is, I think, very interesting. And the fact that the responses to those things come on in the apical dendrites over the course of multiple sessions suggests that they start to carry information about these prediction errors after the circuit has already kind of learned to adapt to them and to modulate the somatic responses down for that. And so the things I want to end on really in terms of thoughts for neuroprosthetics, because this is a neuroprosthetics, obviously, conference, and I haven't talked about neuroprosthetics at all, is the following ideas. So first, when you're doing recordings from the brain and you have a neuroprosthetic device, one of the things that I think is potentially important to think about is the idea that a lot of the signals you might be recording may actually relate to prediction errors and to other signals that the brain is actually using to train itself. And if that's the case, theoretically, you could, via stimulation or closed loop operations, harness that in order to promote training for the device and help an individual to better control the device. The second thing that I think is potentially an interesting idea is that one of the sites that you could try to communicate with for the sake of neuroprostheses is actually with the apical dendrites in layer 1. And theoretically, this could be a less invasive means of communication because you could run devices along the cortical surface and probably detect these apical signals. And I think there's actually a lot of information in the apical dendrites, especially related to learning and prediction error signals. And there are other reasons that I say that beyond this data. And so it's potentially an interesting target for those looking at potential sites for chronic implants that don't induce massive immunological responses or cell death or anything like that. So with that, I'd just like to thank my collaborators on this, Joel Zalberg, Colleen Gillen, Jay Pina, Tim Henley, Joshua Benjo, and Tim Lillycrap, and Jerome Lecoq, who ran the Open Scope project that we did, along with his entire team at the Allen Institute for Brain Science. And of course, I want to thank the funding bodies who helped me do all my work, Healthy Brains for Healthy Lives, NSERC, and CFAR. And thank you very much for listening.
Thank you, Richard. So I guess we have a student. Marco, help me because I'm lost.
Yes.
About
yeah, I'll be handling the student questions for this period. So thank you for your talk. I had a question of my own. Forgive me if I kind of missed it. In terms of the inverse response in your apical dendrites, I'm not sure I understand the physiology as to why. Why is it? Why is there that delay in the learning process?
Well, as to why there's the delay, I can't answer that for you.
Okay.
That's unknown. As to why there can be a segregation, the answer to that is, I think, one of the most interesting components of the neocortical microcircuit. And that is that the apical dendrites, because they are very long, so here's an image of a layer 5 pyramidal cell filled with the fluorescent dye. And these apical dendrites stretch, pretty incredible distances for a cell. We're talking hundreds of microns. In the human brain, I'm not even sure what it might be closer to a full millimeter. And the apical compartments as such are so distant from the somatic compartments that they are in fact electrotonically segregated. Inputs to the apical dendrites can often not propagate down to the somata at all. And evidence from Matthew Larkham and a few others suggests that the thing that enables the apical dendrites to communicate with the somata is the presence of nonlinear calcium channels in the apical shaft that will occasionally induce these particular and unique activity responses called plateau potentials, where there's a long depolarization of the apical dendrite. And that can induce bursting in the cell body, which may relate to learning as well. So what that means is that you can have a situation where a lot of the activity in the apical dendrites doesn't get communicated down to the somata and is in fact fully segregated from the somata and only occasionally gets communicated down as a result of these nonlinear events. And that's what can help you to have this distinction between the responses in the dendrites of a neuron and its somatic compartments.
Thank you. I think that Marco B had a question too.
Yeah, actually, I was very curious after having seen the results about the actual layer 5 recordings that you have. Do you think that this adaptation over time is more to relate to like learning, to attention? Like, does attention play a role in that? I mean, considering the amount of like a repetitive task usage that the animal is getting?
Yeah, it's a good question. So, you know, we can't, unfortunately, so one of the stipulations in the open scope project was that you could only request open loop experiments. So we could not have stimuli that responded to the animal's behavior. And as a result, there was no task for the animal to do. It was just passively viewing these stimuli. And because of that, we can't really say whether the animal was attending to the stimuli or not. So it's entirely possible that, you know, some of these changes reflect ultimately changes in how the animals were attending to the stimuli over time. Perhaps during the first parts of the session, they did or didn't, and then that changed to them attending to it by the third session. So I think it's an interesting question. My personal instinct is that the animals were probably not attending much to any of this. I think they were probably just like whatever. So the fact that we were seeing these responses suggests, in my mind, that it might not be an attention mechanism, given that the animals weren't engaged in a task. But I can't say that for sure.
We had a question from an attendee who asked, is the event recorded so we can have it after finishing?
Yes, I also think we have a raised hand by John Krakauer.
Yeah.
I would prefer answer like that kind
Yeah.
of question on the QA tab.
So are you giving him the mic or?
Yeah, I mean, he should be able to.
Hi.
Okay.
Hi, Blake.
Hi, John.
I have a question. I mean, obviously, if I understand it, this is trying to sort of get implementational granularity for sort of predictive coding kind of hypotheses, right?
Yes.
Sure, right, which is fine. But I wanted to know how one distinguishes something more computationally attractive like that from just sort of adaptation to a perturbation. In other words, you know, if someone sort of tickles you from behind, you feel it, whereas your clothes are actually responsible for far more than that, but you don't attend to it. Force field adaptation experiments, you start feeling the perturbation, and then after time, you no longer feel it, even though it's present. So why isn't this just a lower level adaptation to a perturbation type situation, rather than imbuing it with a much higher level of computational significance?
Yep, that's a great question. So I have a couple of answers to that. The first is, so we designed the stimuli in such a way that we were trying to minimize that potential adaptation. And in particular, if I can get back there, maybe it will be too long, but I'll just use this one. Because the animals were getting different orientations and different random locations, they were not repeatedly getting slammed with the same low-level inputs, right? So some previous predictive coding studies do exactly that. You play like exactly the same sound over and over again, or you show exactly the same image over and over again, and then you throw in an oddball or you throw in something different. That's not what's happening here, right? They are getting different stimuli every time. It's just that what is changing is the mean orientation that's drawn for the fourth frame. Now, you could say they are for 3 frames getting the same orientations roughly, and then they're getting a different orientation on that 4th frame. So in that way, maybe there is some adaptation to the particular orientation. So the answer that I have for why I don't personally think that this is an adaptation response, but I think that properly picking this apart would require more evidence, is 2 other issues. So the first is that we see this response in the apical dendrites come up over the course of 3 days. So unlike a tickle response, right? Like if someone tickles you, you have that reaction right away, and then it dies down over time as you get repeatedly exposed to it. And that is what we see in the Somata. And I agree with you that a lot of the data that has this kind of pattern to it could theoretically be interpreted as an adaptation thing. Though I would still say, you know, it's interesting that it takes 3 days for this to completely die down. Usually adaptation effects are quicker than that. It's not that it takes you 3 days to adapt to someone tickling to you. But let's even just say for a moment that we don't know whether this is some kind of very long scale adaptation. In the case of the apical dendrites, I would argue it's definitely not that, because they are actually learning over the course of the 3 days to respond more to the thing. And that is in direct opposition to how adaptation usually works in the brain. You don't usually get more sensitive to something. I mean, I guess I shouldn't say that to clinicians, people who study chronic pain, but generally speaking, for sensory neuroscientists, you don't see this increase in the responses over time unless it's reflecting some kind of learning by the system. And then the very last thing I would say is that if I go to the delta F over F responses, so perhaps it's a bit hard to see here, but the latency here is on the order of hundreds of milliseconds. Now, if this was a response that was driven by adaptation to orientation, I would expect that response to come on pretty quickly, because, you know, orientation responses should be in primary visual cortex right away. So we should see those sorts of responses within the, you know, 100 millisecond window that you'd normally see responses in 2-photon imaging. The fact that it takes, say, 200 to 250 milliseconds for these responses to come on, in my mind, is a suggestion that these responses, and especially because they're happening in this case in the apical dendrites, that these responses are actually driven by top-down signals. And so this isn't just some kind of adaptation within the circuit. It is indeed something that the circuit has learned in the course of it attempting to make predictions about the nature of the stimuli it's exposed to. But my answers here have been half speculative, half data-driven, and I think admittedly, as we move forward in the field, we want to keep implementing controls to check that we're not just constantly chasing after adaptation.
Thank you.
And the next question came from the audience. Someone was asking, might stimulation of the superficial layers be less effective as a neuroprosthetic strategy if there's such a separation with the layer 5 somata?
Right. Great question. So the answer to that is, in my opinion, that what you would probably want to use, now this ties into a whole host of theories, but you would have to basically stimulate the apical dendrites in such a way that you can trigger those nonlinear responses I mentioned. So, you know, what we do know, for example, so Matthew Larkham, whom I've already mentioned, has a beautiful paper where they show that, in fact, a mouse's ability to detect a stimulus depends in part upon some of these top-down inputs to the apical dendrites and the activation of calcium currents in these apical dendrites. So I actually think that it could be a very effective strategy for neuroprostheses if you can successfully trigger those nonlinear events. If you can't, then yes, you are basically triggering stuff in the dendrites without communicating it to the somata, and that would not be as good. So it would depend on your ability to get sufficient depolarization to trigger those nonlinear events. The other reason, though, that might be a good strategy to take is because there is some evidence, growing evidence, from the McGill lab, from the Bruno lab, and also from computational work from myself and Walter Sen, to suggest that, and Richard No, to suggest that the apical dendrites may be a critical site for driving learning in circuits. And so if you could communicate with the apical dendrites, you might actually be able to guide learning in the neocortical microcircuit. And this is kind of the point that I had ended on here, in part, that it might be a way for you to actually tap into the brain's inner plasticity system and not just rely blindly on the brain's ability to adapt to your neural prosthesis, but to actually have a guided learning strategy within the brain.
I can actually follow up. I actually think that this is a main point on when we're in the field of using cortical stimulation in a very agnostic way, actually, as boringly a term that Dr. Okonko presented the first day, to repair and recovery without knowing exactly what's the mechanism behind, but just knowing that the motor cortex will have somehow to change in order to promote this recovery. This is actually, I think that some of these mechanisms are very much present there, because even when you don't have, so the approach doesn't have any selectivity, at least as far as the approaches that we're using right now. There are so many problems in terms of what fibers are you recruiting. It's a bundle that has many more problems even than like a peripheral nerve stimulation, for instance. But then you activate this bundle, and because you are close to the area which is the final target of these projections, then you are actually actively tapping on that. And I want to try to make this more like a question than than mixed statements. But like I was wondering, actually, when rather than stimulating, we are trying to record. Thinking about all the brain-computer interfaces, trying to get information from layer 5. You suggested clearly that signals that relate to error potential have a big impact. What I saw in your presentation is that the level of adaptation, even just in something that are extremely simple, is enormous. But that there are some reliable features coming from the outside that you might actually be like, because they're different, you're able to capture very well. What do you think are some reliable features that tell you the control that you're having right now, or the kind of recordings that you're having right now? If you see this kind of things happening, this is a clear indication that the brain is trying to have a different strategy, and you should try to compensate to that.
Right, that's an interesting question. I suppose, you know, if I kind of understand what you're driving at, what's interesting is the possibility that you could detect, possibly by virtue of the signals you record, whether the brain is basically detecting unexpected events with the neuroprosthetic device. So if you have a brain-computer interface and the animal is trying to do something and there's an unexpected outcome from it, you would potentially see clear signals about this in the animal's brain. There's actually, for people who do motor cortex stuff, there's a great paper from Adam Hampman and someone else who, the names I'm forgetting, but from Adam Hampman's lab in part, where they do 2-photon imaging recordings in layer 2, 3, and layer 5 in motor cortex in mice during reaching tasks. And one of the things they show there is that, interestingly, they actually have in that data evidence that layer 2, 3 in particular is encoding information about the outcome of the reach, so whether the animal was successful or not. And I suspect that there might be a variety of signatures that we could develop, a sort of bank of signatures over time for neural responses that indicate when the outcome of things were as the animal expected and when they weren't. And that this could be a way of determining whether your neuroprosthetic is properly hooked up to the system or not, or whether additional training has to occur.
Yeah, thank you very much.
Marco, we have a question from the attendees.
Yes.
I guess they cannot ask, no? They need to type.
Well, they could tape on the question and answer panel, and then we can have them answered.
Yeah.
Yeah, sorry about that. But we
2 minutes.
Yeah, we invite you to do that if you want to try to interact. Also, so consider, okay, you in your presentation you actually presented like a focus on apical dendrites as a less invasive means of communication. What you're doing that in a very specific way, which is not very human friendly at the moment.
Oh yeah. You wouldn't want to do this with 2-photon imaging in humans, obviously. So I think what's key is that there's some evidence that you can detect apical dendrite signals from the peel surface just with electrodes on the surface. And that's the key point, is that you could, my understanding, but I'm not a neuroprosthetics expert, so if what I'm saying is bullshit, anyone who's an expert, please ignore. But my understanding is that you could have a non-invasive device laid on top of the peel surface and detect these signatures of apical dendrite activation.
Yeah, so I think that actually the question that just popped up is related to this specific thing, Mimosa.
Yeah. So there was a question that was saying, do you think that neuroprosthetics could focus exclusively on pyramidal dendrites, or would they also need to focus on inhibitory neurons? Because they're thinking of models like the Sacramento model from the Sen Lab that suggests that inhibitory neurons would work together with those apical dendrites.
Right. I mean, so I think, in some ideal world, obviously, we would have bidirectional control over every neuron in the circuit, and great. But in a more realistic scenario, I suspect that in humans where you don't have the ability to genetically target those cells, so barring some kind of discovery of viral approaches that allow for targeting of interneurons in human beings without any kind of genetic manipulation in the subject, probably it would be hard, next to impossible, to record from the interneurons specifically, because they're so few and because they're relatively small. The only ones that might not be the case for are some of the interneurons in layer 1, but like the VIP cells, but they're also pretty small and there's not very many of them. So though I think it would be great to be able to record from the interneurons, I think potentially the most realistic strategy would be to try to focus on the pyramidal neurons. And because they're likely the sort of workhorses of the information processing in the system, even though the interneurons are undoubtedly key, and I personally think they're key to understanding the circuit and the computations that occur in it, in terms of just communication with the circuit, it might be sufficient, I think, just to talk, as it were, to the pyramidal neurons.
Thank you very much, Blake Richards, for accepting our invitation and talking today. We are heading for a 15 minutes break, but before that, I would really like to thank all the public. We have a great, a participation that exceeded our expectation in the past 2 days, with more than 500 people come and join us. During these 2 talks, we had about 280 participants live on screen. This is not something that you see from the audience, but to us, this pays back for a lot of work that we put into this conference. In 15 minutes, yes.
So just one thing, Richard, there was still one question from an attendee on the Q&A. If maybe you can type your answer.